So we're going to be looking at viruses today, and we're going to start first with their structure. So hopefully y'all remember a virus is not overly complex structurally. It's going to consist of a capsid. Remember, your capsid is going to be made of proteins. Okay, so this is a protein-based shell, for lack of a better word, that's going to surround the genetic material. Okay, the genetic material could be DNA or RNA. Okay, so it can be either kind of nucleic acid, either which can be single or double-stranded. Just because the virus contains DNA does not mean it has to be double-stranded DNA. It can be single-stranded DNA and it can be double-stranded RNA. So it contains DNA or RNA, both of which, like I said, can be single or double-stranded. The capsid that surrounds that genetic material, again, is made out of proteins. Okay, the subunits for the capsid are what are called capsomeres. Okay, and those are the little subunits that are going to make up the capsid. Occasionally, you may have a virus that is also enclosed or surrounded by an envelope. That would be on the outside of the capsid. Okay, the envelope is not found on every virus. It's only on certain viruses. Okay, it is usually a phospholipid membrane because it came from the host cell as part of the host cell's cell membrane. So it's going to contain phospholipids and may contain glycoproteins as well. Remember, those are those surface markers. So the viral structure is significantly less complex than a cell, whether it's a prokaryote or eukaryotic cell. There's also a wide variety of viruses. On the upper left-hand corner here, you can see this is a bacterial cell, and all these little things here, these are viruses that are attacking this bacterial cell. So this can help give us a perspective on size of a virus. In particular, compared to a bacterial cell, you can see how significantly smaller the viruses are as opposed to the actual bacterial cell. You can see on the bottom a variety of viruses here. Some have DNA, some have RNA. You can see the capsomeres on this one. Okay, you can see the glycoproteins on others. Okay, you can see that we've got a variety of shapes here, okay, from circular to the geometric shape to the bacteriophage, which is, remember, the kind that only infects bacteria. You've got the rod-shaped viruses. So a wide variety of shapes here and components, both with the genetic material as well as the capsid. Okay, so let's do a little overview of how the virus is going to reproduce. Okay, remember, your virus is going to first inject its DNA or RNA, inject its genetic material into the host cell. Remember, viruses require a host to reproduce. They cannot reproduce on their own. And basically, they need to get their genetic material into the host so they can use the parts of the host cell to manufacture themselves. Okay? That they can assemble their own, they cannot assemble their own proteins. They're going to need the host cell to do all of that for them. So the first thing they'll need to do is inject their material into the host cell. That genetic material then can be replicated. Okay, so whether it's viral DNA that's copied or messenger RNA that's copied, the genetic material gets replicated, and it will need to also be transcribed into messenger RNA if it's in the form of DNA. If viral DNA is, the virus contains DNA, then that viral DNA is going to have to be at some point transcribed into messenger RNA so that the proteins can be made for the capsid. So we've got the transcription and the manufacturing of the proteins that are going to form the capsid, and then those things will assemble back into adult, for lack of a better word, viruses, active viruses, viruses that can go out and infect other cells now. Remember, when the virus explodes out of the cell, okay, the cell has been lysed, okay, or it's gone through the process of lysis, and that's that bursting. And remember, that's going to destroy the host cell. That's how viruses make us sick, is by destroying the host cell through reproduction like that. Okay, viruses that only reproduce this way, by destroying the host cell, those are what are called virulent phages. Okay, they reproduce via what is called the Lytic Cycle. Okay, and the Lytic Cycle is the one that destroys the host cell because those new viruses explode out of the host cell. Okay. Some of your bacteria are going to be able to defend against these viruses. Okay. Not all of them, but some bacteria have what are called restriction enzymes. So again, we're only talking about prokaryotes here. Okay, so some of your bacteria will have restriction enzymes, and restriction enzymes are able to cut DNA. And so they cut DNA, they look for certain DNA sequences, and they cut DNA at that particular spot. And so some bacteria have restriction enzymes that'll basically end up cutting out the viral DNA. So when the virus enters the host cell and its DNA mixes with the host cell DNA, some bacteria can use these restriction enzymes, and they'll find these DNA sequences that'll bookend the virus DNA, and when they cut there, they end up cutting out the viral DNA, which would allow the bacteria to survive the viral infection. Okay, let's look at this lytic cycle a little bit closer. And we're going to be following what's called a bacteriophage through this. Okay, remember, bacteriophages are viruses that infect bacterial cells only. Okay, so they infect prokaryotic bacterial cells. So the first thing that bacteriophage, so here's my bacteriophage right there, the first thing that it's going to need to do is it's going to need to attach to the bacterial cell. So the phage is going to use receptors that are on the outside of the bacterium, okay, and it's going to bind with the receptors on the outside of the bacterium, and that's how it's going to be able to actually attach to the host cell. After that attachment occurs, the phage is then going to need to release its genetic material into the host cell, okay, and so it will inject its genetic material into the host cell, and the host cell's DNA is going to get broken down. Got to break down the host cell's DNA so that the host cell is only using the virus's DNA at this point. So once the virus's DNA is inside of the host cell and the host cell's DNA is being degraded, then the virus is going to start synthesizing. It's going to start using the host organelles. It's going to start using the host ribosomes. It's going to start using the host cell's parts to break down, to start making its own proteins. So in this case, it's a prokaryote. We've got a bacterial cell that's being infected. So the virus is going to be taking over its ribosomes so that it can, and using its parts, using its amino acids, using its tRNAs, so that it can produce its own proteins. It needs its own proteins to make the capsid. And so it's going to use these to be doing transcription as well as translation. So the nucleotides that are needed to make the messenger RNA, the tRNAs, the amino acids, all of these things the virus is going to be taking from the bacterial cell to be assembling into its own proteins. From there, the virus will start assembling those finished pieces. Okay, so you can see over here on the left, okay, it's encapsulating the genetic material into the capsid. So then we have a complete capsid that contains genetic material, assembling the tail and the tail fibers to produce a complete phage. So now I've got this bacterial cell, this host cell, that is full of these viruses, okay? These fully assembled, complete, ready to go infect other cell viruses or phages. From there, those new viruses are now going to release or lyse the cell. And so when these viruses explode out of the cell here, this kills the host cell, okay? And so, again, that's how viruses cause infection in us. They're killing our cells as they're releasing out of our cells, okay? The cell has exploded, okay? And these phages then are ready to go infect other cells. Remember, these will be usually virulent phages that are going to go infect something else and do the lytic cycle, okay? They're not going to go sit dormant somewhere. The other option a virus has is to go through what's called the lysogenic cycle. And so in the lysogenic cycle, the virus is dormant. Okay? It is not using its genetic material to make new viruses. Okay, its genetic material is being copied, but that genetic material isn't necessarily on. It's not making new viruses. It's not transcribing and translating that genetic material into the proteins, into the capsids. And if you look at the right side of this diagram here, that's where you'll see this lysogenic cycle taking place. Okay, so let's start up here. Okay, with the phage is infecting the bacterial cell. And so it's injecting its DNA, it's injecting its genetic material into the bacterial cell. And it has two choices. Okay? If the lytic cycle is induced, then it will go this direction, and it will go through the assembly and the lysis phase. If the lysogenic cycle is entered, the viral DNA, you can see the blue there, gets incorporated into the host cell DNA. So it becomes part of the host cell DNA. And when it does that, that is what's called a prophage. Okay? And so the prophage is this combination of viral DNA and host genome. And so then every time this bacterial cell reproduces, that viral DNA gets copied because it's now part of the bacterial genome. And so every time the bacterial cell reproduces, it copies its DNA. So the viral DNA is being copied, but it is not being used for transcription and translation. Phages that do this are what are called temperate phages. Temperate phages can do either lysogenic or lytic cycle, different than a virulent phage that only does the lytic cycle. Okay, temperate phages, like I said, can go be dormant in this lysogenic cycle. Now, the virus can leave, the genetic material can leave the chromosome. Okay? It can leave the host cell chromosome and initiate the lytic cycle. You know, if your immune system is weak, if it can tell that it would be able to survive and start translating and transcribing, then it can leave the chromosome and it can initiate the lytic cycle. A little comparison here's some contrasting between the lytic and the lysogenic cycle. Okay, so first of all, they both have to have this injection has to happen. The phage has to inject its DNA genetic material into the host cell. The lytic cycle here can be done with a virulent or a temperate phage. Lysogenic cycle is temperate phages only. During the lytic cycle, the host DNA is going to be destroyed. During the lysogenic cycle, the host DNA is not destroyed. The viral DNA is mixed in with the host DNA, becoming that prophage, so that it keeps getting passed on. And so it's not doing transcription, it's not doing translation, like it is in the lytic cycle, where it's actually making new phages. And in the lysogenic cycle, no new phages are being made until it leaves the chromosome and starts that lytic cycle. Some of these viruses, as they exit the host cell, will form an envelope around them. And that envelope comes from the host cell's membrane. They use the host cell's phospholipid bilayer to make this membranous envelope. You can see as this virus here is exiting the host cell, it's taking some of the host cell membrane with it. And so now it's enclosed. This is its envelope here that it's enclosed in. And so it's the membranous envelope of the virus, like I said, came straight from the host cell. And so what this does, is it offers the virus protection from the immune system. Your immune system isn't right away going to go attack this virus because it thinks it's a cell that belongs to you, because it is wrapped up in the host cell's membranous envelope. So a virus having an envelope on it is usually beneficial to the virus, helping to protect it from the immune system. Okay, so let's look at some viruses that are going to attack or infect animals. And so viruses that infect or attack animals, they have the widest range of RNA genomes. A lot of the viruses that infect and attack animals contain RNA for their genetic material. And these include a category of what are called retroviruses. An example of a retrovirus would be HIV. That is the most common example here of a retrovirus. So a retrovirus contains an enzyme in it. It's called this enzyme here called reverse transcriptase. I know me writing it is not much better than the small print there, but now you can have two bad examples. So reverse transcriptase, remember there's my ASE enzyme. Reverse transcriptase is going to do exactly what its name says. Usually we do transcription, we take DNA to RNA. Well, we're doing reverse transcription. So we're going to take RNA to a DNA. So this virus, when this virus attaches to the host DNA, the host cell, it's going to inject in it the reverse transcriptase enzyme and its genetic material, which is RNA. And so you have this viral RNA that's inside the host cell and the reverse transcriptase enzyme. The reverse transcriptase enzyme is going to be able to convert that viral RNA into DNA. So I have a strand of RNA and the red on the top there, and a strand of DNA in the blue on the bottom. Okay, and now then the virus can use the host cell's materials to then make the second strand of that DNA. So we make the complementary strand to the original. So we did reverse transcription here, went from RNA to DNA, then made a complementary strand of the DNA. So now I have a double-stranded DNA, double-stranded section of DNA that came from the virus. The virus is then going to incorporate that strand of DNA into the host virus. Okay, you can see that happening right here. Okay, that is called, again, a provirus, just like it was a prophage before with the bacteriophage incorporating into the host genome. Now we have a provirus, and so that provirus contains a portion of viral DNA as well as the host cell's DNA. And so now that viral DNA is either being replicated when the cell reproduces, okay? So it's either being replicated or it's being transcribed. And if it's being transcribed, it's providing RNA for the next group of viruses, okay? And it's providing RNA to produce the proteins that it needs for the capsid. So it's providing RNA that can be translated into proteins, and it's providing the viral genetic material for the next protein, for the next set of viruses. But that viral DNA is also now still incorporated into the host cell DNA. So now these new viruses will assemble and lyse out and be able to go infect other cells. And the original host cell, it didn't die, but it is still infected because it still contains that viral DNA that is incorporated as a provirus into the host cell DNA. We also have viruses that can infect plants. Most of these are also an RNA-based virus with an RNA genome. Okay, there's two ways, really two basic ways that viruses can be passed from one plant to another. There's what's called horizontal transmission and then vertical transmission. So with horizontal transmission, the virus is going to be transmitted basically from cell to cell, through the cell wall from cell to cell. With vertical transmission, or vertical is up and down, vertical transmission, it's going to have come from the parent. So horizontal transmission, it's going from cell wall to cell wall. Vertical transmission, it's going from parent to child. We also have almost like a subcategory of viruses, which are called Veroids, and then we'll also talk about what are called Prions. Okay, so Veroids are small little RNA, pieces of RNA, so small circular RNA molecules that can replicate inside of a host cell, but they do not encode for proteins. Okay, so these do not encode for a protein, so it's like half of a virus. There's genetic material here, there's RNA here, but there's no protein surrounding it. These have a major impact on our plants. Okay, these disrupt the regulatory systems, in particular of growth for plants. So they disrupt the regulation of plant growth, and so they are infectious to plants. But again, there's no protein here, just this little circular RNA molecule that gets inside the host cell, it replicates, it disrupts the plant system, but it is not a full-fledged virus because it doesn't have all the parts of a virus. On the other side then, we also have what are called prions. Prions then are infectious proteins. They are not, there's no RNA here, okay? There's no DNA here, there's no genetic material. So again, it's the other half of the virus, it's the protein half of the virus. So these are infectious proteins. These cause brain disease in mammals. Okay, you've heard of the diseases these things cause. Okay, these proteins are going to reproduce, we use that term loosely, by changing regular proteins into these infectious ones. And if you look at this picture at the top here, your normal proteins here, your rectangles, and these chevron-shaped ones are your prions. And when the prion comes in contact with the original protein, it turns it into an infectious kind. It changes the conformation of it, and so you end up with these large aggregates or groups of infectious proteins. And so these proteins, like I said, there's no genetic material here, it's just a protein. These cause things like mad cow, scrappy, which is basically mad cow in sheep, brain disease in sheep, and what is called Creutzfeldt-Jakob. Creutzfeldt-Jakob is in humans. And so all of these things, basically these proteins turn the mammal's brain into a sponge. They create holes in the mammal's brain, it becomes soft, and it has these holes in it now, and so they're usually lethal relatively quickly. Okay. So we'll be working with a little bit with viruses in class, and kind of going through again with some models on how they are able to reproduce, and taking a closer look as to whether we think then that they are alive or not.